Excellent. All right. Well, today I'm going to be talking about genetic variation of lake whitefish in Lake Erie. Full disclosure, I am not a genetics guy. Peter was going to try to present this, but he could not. So we had to go deep into the bench. So bear with me. I see Amanda is here, so she can correct me when I say something incorrectly. But yes, to jump into it, I just wanted to give a little background on the importance of lake whitefish in Lake Erie. Obviously, they support commercial fisheries, especially in the upper lakes in present times. Historically, millions of pounds per year harvested out of lakes Michigan and Huron. But when you look through the history of Lake Erie, they also used to pump out quite a few. The late 1800s, there was these expansive pound net fisheries across the entire West Basin. And one of the first commercially pursued fish at that time was whitefish. And if you read some of the early writings from really the late 1800s into the early 1900s, at one point there were 3 hatcheries in the West Basin that were trying to kind of rebuild the whitefish stocks. And that I believe they were stocking fry at the time. So way back in the day, they were hugely important. The fishery somewhat collapsed then. They became less economically viable. And then the fishery switched to Cisco, in which that was just insane as well. Up to 50 million pounds a year, I think, was peak harvest in 1918. And that would be equivalent to basically the combined harvest across all the Great Lakes of all species in contemporary times. But anyways, I'm getting off track. So they were historically an important fishery in Lake Erie. Back in the day. Obviously, they're also an important part of the food web. They occupy mid-trophic levels. They connect the benthos to kind of the upper zones in the water column. And so an important part of energy transfer. And as subadults, they're an important prey fish for large predator species. Lastly, they're widely viewed as an indicator of ecosystem health. They require cooler, well-oxygenated water and kind of a stable food web. So, next slide. Yes. So historically overfished, as I kind of already got ahead of myself. See, I'm totally winging it and not following my notes or the slides, so I apologize. Yes, historically overfished in the late 1800s, and then Cisco came into the picture, and then the Cisco population crashed in 1924, and then we went back to whitefish. And whitefish harvest peaked at around 7.7 million pounds in the late 1940s, which I tried to kind of put that in perspective compared to the other lakes that still support fisheries today for lake whitefish. And it seems like it's 7.7 million pounds is kind of a lot for a lake the size of Lake Erie. And so after that, the population crashed and harvest was really low for quite some period of time. And then in present times, there is some harvest. There's just not a ton in present day. And they're currently managed under the assumption that they're a single stock and a single genetic group, but we really didn't know that going into this. And this picture here is kind of cool. This is the Detroit River. This is a holding pen, I believe, on Grassy Island. And during the peak of the whitefish fishery there, they would harvest fish, I think, via pound nets or maybe even staked gill nets. And then they would transfer them to these holding areas in the middle of the river, kind of where the currents circulate well. And then they would just harvest them as needed so they could always have fresh fish at the market. And at the peak of the Detroit River fishery, you know, the estimates are not always super certain because it's, you know, late 1800s, early 1900s. Up to 500,000 pounds a year just out of the Detroit River is kind of the best guess. The Detroit River as a whole was pumping out up to 3 million pounds a year, and that was mostly walleye and lake whitefish. The Detroit River, basically, that population crashed when they cut the Livingston Channel and the Amherstburg Channel and destroyed a lot of the spawning habitat. But anyways, sorry, this is not supposed to be a history lesson, so I apologize. The objectives of this study were to really kind of get at the question of, is this a single genetic stock or is there some genetic substructure? And so to do this, we thought the best way to assess this would be to gather these spawning adults from spawning grounds as they're spawning. Catch them in the act, if you will. Just bust the door open and net up some whitefish, because we thought, they kind of have this life history trait where they will migrate to the west reefs and they'll spawn there, and then as the water temperatures warm in the summer, they have to shift to the East Basin to get adequate oxythermal habitat. So if we're going to try to get them in the summer months, it's probably kind of a mixed situation, whereas if we get them on the reefs, maybe there'll be some substructure going that route. And so, yeah, that was the main objective. Okay, are there genetically unique populations associated with these spawning aggregations? And then a second thing that came out of this is we could use whole genome sequencing to explore effective population size through time and kind of just track back and have a retrospective look at how the population may have changed in light of some of these anthropogenic stressors like fishing. And so to get into the sampling, here's a great picture. And so the technicians in my office kind of hate me because I'm like, oh, it's not, blowing 30 miles an hour today. Let's go out to the reefs. Let's bust through some ice. And so our sampling for this project was primarily late November into early December. These were overnight gillnet sets on reefs, and then some exploratory gillnet sets in the Detroit River and then over by Fairport Harbor. And then we also cooperated with some commercial guys in the West Basin. They were super helpful. They got us some fish from in the vicinity of West Sister Island and Maumee Bay and some of the West Basin reefs. So it was kind of a cooperative effort across the board. And so to dive into the methods, we used two approaches. We used GT-seq panel that had already been used in Lake Michigan, and this was basically based on a small set of specific genetic markers across the genome. And then we did whole genome sequencing, which obviously is a little more comprehensive, hence whole genome sequencing. And then that enabled us to kind of do this retrospective analysis of effective population size. So to get to the fun part, we were able to get 371 lake whitefish. We did have representation from all three basins and from the Detroit River. The East Basin, I'm so thankful for Tom McDougall for working with the fishermen over there and getting us a significant sample during November and December, because that side of the lake is straight up violent that time of year. So that was great. We did exploratory netting. My office did in the Detroit River and in the Central Basin, and it was a grind. In the Detroit River, we netted for, I think, 6 weeks straight, 4 days a week, and we ended up catching three lake whitefish. They were all at the mouth of the Amherstburg Channel. They were all males. Some were expressing. They were, if they weren't flowing, they were close. So they're spawning somewhere near there. We never could pinpoint exactly where. Andy Cook, another of our Canadian partners, he was doing some tagging over there and got us, another 12 or so from around Fighting Island. So we had something like 15 total from the Detroit River. Not as many as we had hoped for, but it's better than nothing. And then we also did some netting at Fairport Harbor. This was based on some of the early life history work from other folks that have been looking at eggs and larvae. And they had found whitefish eggs there. And so we did some sampling there, and we were able to get just 6 from the Central Basin. But we did have, I think, somewhere around 50 from Long Point. So pretty good representation way over there in the East Basin. And so for the GT-seq, we ultimately used 331. We threw in, I'm going to now mention that as part of the analysis, we did incorporate a spawning group from Lake Huron from Coreyon Reef. This was kind of to act as like a control or an out group. But then also we wanted to see, is there a genetic difference between these Lake Erie fish and the Lake Huron fish? Because we do see evidence of fish obviously coming down from Lake Huron through St. Clair and through the Detroit River. And so when we looked at it using GT-seq, we saw that everything in Lake Erie diverged from Lake Huron. Okay, seems like it's working. but there was really very little separation from the Detroit River all the way over to Long Point Bay. Not much going on, and I'll show some figures here to demonstrate that further. For the whole genome sequencing, we used a smaller subset of that. That's a little more time intensive. Similar patterns observed. In essence, we saw no genetic difference between those Detroit River fish and those Long Point Bay fish. And yeah, I'm not going to spend a whole lot of time on this because this is just probably too much information. But these are FST estimates. The GT-seq data is kind of in the top right quadrant, or I don't know, half, if you will. And then the whole genome is the bottom left. And again, this is just kind of reiterating that the Huron fish, Coreyon Reef and red, diverged from everything in Lake Erie. And then within Lake Erie, there's very little genetic separation. And here's PCA biplots, kind of using the same data. As you can tell, there's more points up here at the top half. Sorry. That's obviously the GT-seq, where we did all 331. And then down here is the whole genome sequencing. The gray crosses, that's the Lake Huron fish. And they're kind of separating out nicely. And then we have it color-coded by spawning aggregation, and it's just kind of a shotgun blast of points. There's not a whole lot of separation there. Now, with the whole genome sequencing, there's kind of these interesting outliers. I wouldn't read into that too closely, because if you look at how much variation is explained at each axis, it's not a whole lot, but there were some outliers, and those were those East Basin fish. Take-home message, though, not much going on in terms of genetic substructure. So caveats. Obviously, in November, December, you have very limited windows of opportunity to get out there and survive, which is important. And we didn't get every single spawning aggregation, clearly. I'm sure there's plenty more that are occurring all over the lake. It's a huge lake. It's the 13th largest lake in the world. But what we could say fairly conclusively is that all these fish in the west and these fish in the Detroit River are very similar to these fish over here, and they're 250 kilometers apart. So could there be other populations in Lake Erie that are genetically distinct? Maybe, but it doesn't really seem very likely given this relationship here. But, so that was kind of rather uninteresting. The retrospective analysis of effective population size was kind of fascinating. This was based on the whole genome sequencing data, and it's based on, I think Amanda would know better than this, but it's a linkage disequilibrium between SNPs across the genome is used to calculate effective population size through time. What we ultimately saw was this big bottleneck 20 to 25 generations ago. And if, from what we've seen in a lot of our whitefish in the West, sexual maturity is 3 to 5 years, that would be 60 to 80 years ago, 60 to 90 years ago, something like that. You start to see this crash, and that corresponds really nicely with the fisheries crash when harvest peaked at 7.7 million pounds, I believe, in 1947, and then just kind of fell off completely after that. And so, yeah, there's a lot going on here. This is the effective population size through time, but I just want you to focus on the solid red line here. That's Lake Erie. And as you go right along the x-axis, you're going back in time. And you see, if you go back 20 generations to around here, you see this big drop off. Effective population size here was estimated at 30,000, and now it's 6,000. So it's like a 5-fold decrease. Obviously, effective population size is not the same as census population. But, there's clearly this big bottleneck going on at that time. Well, let's look at the lake whitefish harvest data from 1867 to 2025. Look at this. This is I believe, maybe 1950, something like that. It peaked at around 7.7 million pounds and then just crashed. And that's within that, 60 to 80 year window that corresponds with that collapse in effective population size. This is just kind of a teaser. You'll have to read it in the paper. We have a paper that's going to probably be out here in the next couple of months. There was some looking at kind of adaptive variation in parts of the genome that may be under selection using some of the whole genome data. And we saw some cool things potentially going on in both Long Point Bay and Maumee Bay whitefish, but we'll save that for the paper. So in conclusion, minimal genetic substructure in Lake Erie. We didn't get maybe as many fish as we would have liked in the Central Basin and the East Basin, but we do have samples that are 250 kilometers apart, and they are from November and December when those fish are actively spawning, and we're not seeing anything there. So yes, there may be populations out there that could have some genetic substructure, if you will, but we did not see it. And then the kind of cool takeaway here was looking at effective population size through time. We did see a big crash that was correlated with that 20 to 25 generations ago, which would have been kind of the peak of the commercial harvest. And so I think that's it. So sorry it was, I don't know, would you give me 30 minutes? I think it was only a 17-minute talk, so I apologize for that. But thank you, guys. I'll take any questions.